All right, welcome back, fellow scientists. So we are going to dive right back into our discussion of biotechnology. In this video, we are going to look at two more tools and two more processes. So we already talked about our first tool, restriction enzymes, in the last video. Our second tool is called polymerase chain reaction. It's affectionately known as PCR, right? And its job, it simply makes millions of identical copies of DNA. Now, you'll remember DNA polymerase all the way back from chapter 12. It's used in DNA replication. So you have a single strand piece of DNA, and then it adds A's to T's and C's to G's. So it makes a polymer. That's why polymerase. It's an enzyme ACE. So hopefully these names are starting to make more sense, and you're starting to become familiar with them. So it makes millions of identical copies of DNA. How does it make millions of identical copies of DNA? Well, it's really very simple, actually. So we start out with our one piece of DNA, okay, and then what we actually do is we heat it. So it's in a little mini centrifuge tube, and we put it in a machine, and it's in like a aqueous solution, a water solution, and whatnot. So we put it in a machine. Machine raises the temperature. It doesn't boil the water, but it raises it. And so what that heat does, if you remember back from chapter 2, heat denatures proteins, so it changes their shape, right? Heat also denatures DNA. So no longer is this DNA double helix, but the two strands of DNA, they separate. They denature, that breaks the hydrogen bonds, and so now we have two single-stranded pieces of DNA. So we have this special DNA polymerase, which I'll talk about in a second, that comes through, and it says, oh, we have single-stranded DNA. So I'm going to add A's to T's, C's to G's up here. I'm going to add A's to T's, C's to G's down here, and then all of a sudden we've just doubled the amount of DNA that we have. Well, what if we did that for another cycle? So we heat it up, so now we have four single-stranded pieces of DNA, and then our DNA polymerase is going to come along, A's to T's, C's to G's. So with each round, we double the amount of DNA that we have. Right now, I know what you're thinking. Mr. Leo, you said heat denatures proteins, and we use heat to denature DNA, but DNA polymerase is a protein. It's an enzyme, so why doesn't that heat denature it? Well, scientists are kind of smart that way. They found a special bacteria that lives in like the hot springs at Yellowstone National Park and other places down deep sea hydrothermal vents, things like that, that can withstand that kind of heat. So it's called Taq polymerase after the bacteria that it was found in, right? And so because this bacteria lives in very hot environments, it has to have a DNA polymerase that can withstand that kind of heat. So scientists said, oh, well, that's kind of cool. We'll use that for this. And so now we can heat stuff up so that the DNA denatures, but we're not denaturing our Taq polymerase. We're not denaturing our special polymerase. So that's the second tool that we're going to talk about. Third tool that we're going to talk about is gel electrophoresis. Now, this is something that we actually get to do in class. We do it with dyes in biology. We do it with actual DNA in biotechnology and in AP biology. But gel electrophoresis, the whole point of gel electrophoresis is that it separates molecules based on size and charge. Well, Mr. Leo, that doesn't sound very exciting. I know, but that's okay. It will be once you understand the process. So we're going to go through this step by step. So step number one, make gel. Okay, basically you're just making Jell-O. You're adding like a sugar, and then you're heating it to boiling, and then you're letting it cool. And we can put wells. So when we cast our gels, we have a little thing called a comb that we can stick in there, and then the liquid goes around the little tines of the comb, and it makes wells. It makes little indentations in the gel. So we can put our wells to one end if we want to analyze DNA, or we can put our wells in the middle if we want to analyze dyes. So in class, we're going to put our wells in the middle when we make our gel. So step one, make gel. Step two, put the gel in a chamber with salt water because we're going to run electrical current through the gel, and surprise, surprise, electricity does not run through pure distilled water. It has to be salt water, okay? So put gel in chamber with salt water. Number three, load samples into the wells. So up here, I have a sample of DNA from a crime scene. I have a sample of DNA from suspect 1. I have a sample of DNA from suspect 2. Okay. Here I have dyes. So we could say that this, I don't know, is a blue dye, and this right here is a red dye. Okay. So we're going to load the samples into the wells, right? And then 3, we're going to apply an electric current to the chamber. I can't picture that. Oh, here I can. So we have our negative pole over here, so that's black, just like a car battery. The black wire attaches to the negative terminal. We have our positive pole over here, so that's red. So just like car battery positive, the wire is red. So we're going to apply electric current to the chamber, and then we're going to analyze the results. Now here's how we analyze the results. Let's start over here with the dyes. Okay, oops, don't do that. So the dyes. So this red dye right here is, we see that it's moving towards the positive end, so that means that it is a negatively charged dye because opposites attract. Okay, this blue dye up here, we see it moving towards the negative end, right? So that means that it's a positive dye. So that's one way that we analyze the results, right? Because remember, it separates molecules based on size and charge. So that's how it separates it, based on charge. And then also it separates it based on size. So DNA, because of the phosphate groups, is only negatively charged. So that's why it's moving towards the positive end of the gel, right? But DNA isn't all the same size. So this fragment right here stays close to the well. It doesn't travel very far through the gel. It is a very large fragment. These fragments down here, they are further away from the well. They travel faster. All right, if you think about like small things versus large things, small things usually travel faster, not always, but you know, you get the idea. So small DNA travels faster, so they'll travel farther away from the wells. So what we can do, and we'll see an actual example of this here in a couple of slides, but what we can do is we can analyze people's DNA. So we can take the DNA that was left at a crime scene, we can compare it to the different suspects, and we can see that, oh, it looks like suspect 2, we found their DNA at the crime scene. Now, I don't know the crime, I don't know what, that's for a judge and jury to decide, but this is one way to use, or this is one example of how to use our third tool, which is gel electrophoresis. All right, so then let's go on to our processes. So now, what can we do with this? Well, let's say that we want to determine the sequence of DNA. So we got to do something called DNA sequencing. It's a very apt name. Determining the order of bases in a sample of DNA. Now, up until this point, we've just kind of drawn DNA and we've said, okay, A's and C's and T's and G's, and we know the order, and we've just kind of assumed that, right? Well, it's not like we can look at that and see that underneath the microscope. Even a big long strand of DNA is too small to see if it's just single-stranded, or if it's a double helix, but it's just one double helix with even the most powerful electron microscopes. I think recently they actually were able to visualize like a clump of 7 double helixes with a very powerful electron microscope. But that's as far as we got. There's no way that we can actually see the A's and C's and T's and G's. So how do we know what the sequence of DNA is? Well, we do something, like I said, called DNA sequencing. So here's how we do it. We use PCR. Okay, so we make millions and millions of copies. Okay, and then we add. So we put that in 4 different tubes, and then we add different chain terminating nucleotides to the different tubes so that they can't, like DNA polymerase can't physically add another nucleotide to that chain. So then we go along, DNA polymerase goes along, and just randomly it'll add in these chain terminating nucleotides. So let's say that this is the piece of DNA that we're studying. So we did PCR and we heated it up and we were left with single-stranded DNA. Okay, so DNA polymerase is going to come along, and this first one is A, so let's say just randomly it adds a T just right away. Okay, so that's done. That chain is terminated. We're done there. Okay, let's say that on another piece in another tube, the DNA polymerase comes along, adds a normal T, but then adds a chain terminating A. Okay, another piece, another tube. Remember, we have millions of copies because we did PCR AT, and then adds a C, chain terminated. Done. ATC T, chain terminated, done. And these are not only special in that they terminate the chain, we can't add new nucleotides, but they're also special in that they glow, they fluoresce, okay? So then we take all of those and then we put them into a gel, right? Here's our little well. And so this is gel electrophoresis. And so we can see that T, the one with the T nucleotide, chain terminating nucleotide, that's the smallest because it only has one. And so that's going to run through the gel the fastest. So that's going to be down here. A is second fastest, second smallest, so that's going to be down here. C is third, so that's going to be down here. The next T is in spot number 4, so that's going to be down here, so on and so forth. And so that is how we get the actual sequence of the A's and C's and T's and G's. Early 2000s, I think 2001, 2003, this project was actually completed for the human genome. So we now know lots of different stuff about the human genome. The sequence of the human genome, 3 billion letters, basically done this way. So that's our second process, is DNA sequencing. And then our third process, I already kind of talked about DNA fingerprinting. So it's called fingerprinting because it identifies a person based on their DNA. Now, it has nothing to do with actual fingerprints, except for that we used to use fingerprints to identify people from crime scenes. Now we can use DNA fingerprinting, but we're not using anything like their actual fingerprints. We're just using their DNA. Okay, so you take different DNA and you chop it up with the same enzyme, right? Every single person on Earth, except for identical twins, has different DNA. And so if you chop it up with the same restriction enzyme, you're going to get different size fragments, okay? So let's say that the wells are up here, and then we apply the charge, and so then we have lots of small fragments down here and large fragments up here. And we actually get, it almost looks like a barcode for a human being. So these are the fragments that the restriction enzymes made from our crime scene DNA. Here's the fragments from suspect 1. You can see that doesn't really match. Here are the fragments from suspect 2. You can see that actually matches. And here are the fragments from suspect 3. You can see that that doesn't match suspect 2 or suspect 1 or the crime scene. So now, just like the example I gave you before, we can see that suspect 2 was the individual that was actually at the crime scene. So those are our two tools, and two more tools and two more processes for biotechnology. Thank you so much for watching. This has been Mr. Leo, and I will see you on the flip side.